Having told you the story of my brief story on CADASIL, I think it's time to introduce the first speaker. Anne Joutel doesn't need any introduction. Anne, please. Thank you.
So, thank you very much. And first, I would like to thank Marie-Germaine for this great meeting. I would like also to thank Elisabeth Tournier-Lasserve, who is my mentor and who gave me this unique opportunity to start my career in a lab and on this very fantastic disease. So what I would like today is to give you an update on the mechanisms of this disease, and even though we still have a number of gaps in our understanding of this disease, we did learn some things. So let's begin. So at the starting point, the starting point is very simple. I realize the problem with this. Okay. No, it's okay. So the starting, and I need to check my watch. Okay. So the starting point is very simple. You have a single point mutation in this NOTCH3 receptor, which is predominantly expressed in smooth muscle cells, and this is the starting point. And at the end stage of a disease, you have this massive degeneration of the white matter, lacunar infarct, brain atrophy. You have also these vascular deposits of NOTCH3 protein, of gum deposits, and we know that these deposits occur in between. So during the past years, we aimed to address the following issues. On first, what are the earliest cerebrovascular changes? So what I will show you today, we are dealing in the lab, we are dealing with the earliest mechanisms of a disease. So I'm not addressing the mechanisms at the end stage of a disease, but at the very beginning. So what are the earliest cerebrovascular changes? Is the disease caused by a malfunction of a receptor? We have these vascular deposits. So what is the mechanisms of formation of these deposits? And do these deposits contribute to the phenotype? So one of the most straightforward approach to investigate and to determine the earliest cerebrovascular changes, the earliest changes in the Mendelian disease, is to have an animal model. So, so all kinds of genetic manipulation have been tried to recapitulate CADASIL in the mouse. And the expectation that just by introducing a point mutation into the mouse genome will mimic a human form of a disease was not realized. And what all these mouse models develop well is the NOTCH3 ECD deposit and the gum deposits. So we have mice with a single point mutation in the NOTCH3 gene, mice overexpressing a small amount of a mutant protein under a smooth muscle cell promoter, and mice, so only this mouse model, which we generated in collaboration with the Hübner's lab in Berlin, so only this mouse model, which overexpress about 4 times the mutant protein, develop white matter lesions. So what we learned from this mouse model is that these mice exhibit first NOTCH3 ECD deposits, and there is a delay between the appearance of NOTCH3 ECD deposits, so they exhibit these deposits as early as just after birth. Could you switch off the light, please? Yes, thanks. So what these mice develop well, they develop very early NOTCH3 ECD deposits in the vessels. They develop from 6 months of age, they develop the gum deposits, and from 12 months of age, they develop white matter changes. And in the lab recently, Emmanuel Cogna showed that we could evidence these white matter changes using an anti-MBP antibody, and showing a degradation of the myelin, so the presence of myelin debris as early as 12 months of age. And at 20 months, we have this large vacuolation of the white matter tract. So recently in the lab, Emmanuel has examined in detail the pathology by electron microscopy, the nature of these vacuoles. So what he found, very surprisingly, is that the majority of these vacuoles correspond to the accumulation of fluid inside the myelin sheath and not outside. So it's not an extracellular accumulation of fluid. So the majority of vacuoles develop, and the majority of these vacuoles correspond to the accumulation of fluid within the innermost layer of the myelin. And as you can appreciate on this slide, the axon is pushed away. So in a very small number of cases, we can see some vacuoles and some fluids inside some cells, but it's a very minority of vacuoles. So then Emmanuel analyzed the axons, and he analyzed in detail, he did a number of electron microscopy analysis, and what he found is that in the majority of these vacuoles by electron microscopy, and he counted all these lesions, and he saw that in the majority, in 60% of vacuoles, the axon was still present, and only in 40% of vacuoles, the axon was degenerating or absent. And to confirm this result, he used another approach, and he did confocal analysis combining NMBP staining and staining labeling the axons, and he did confocal analysis, and he found that within the myelin debris, as you can show in red, in the majority of myelin debris, the axon is still present or is on the border on the edge of the myelin debris. But again, in the majority of lesions, the axon was still present. So suggesting to us that the axonal degeneration is a secondary event, and on the first event, there is a myelin degradation. So we took advantage of this mouse model to investigate the mechanisms of these lesions. So it has been proposed that white matter lesions could be due to a failure of the blood-brain barrier. And in CADASIL, so keep in mind that we are investigating the earliest phase of a disease. We are not at the end stage of a disease. We are at the beginning, and we are interested in determining what are the mechanisms which initiate the lesion. So we did some extensive analysis of the blood-brain barrier using both an electron microscopy approach, and we did also, and Marie Monet in the lab, did functional studies. So she injected in the mouse dextran, 70 kD dextran molecules, and she did confocal analysis in the white matter, and she delimited the boundary of a vessel using a PKM staining, which labels the endothelial cells. And what she found definitely is that there is not any leakage of dextran outside the vessels. So in this mouse model, we don't have any leakage, and we don't have, so the blood-brain barrier is preserved, and this is very consistent with electron microscopy analysis. So it has been postulated that these white matter lesions could be due to vessel stenosis or fibrosis. So again, at the very beginning of a disease, so we have these white matter changes, and if you look into detail to these vessels, both at the pile arteries and also at the intraparenchymal artery, there is not any fibrosis, not any smooth muscle cell degeneration. So we have a mouse model with white matter lesions, no alteration of a BBB, no stenosis of the vessels. So what is wrong? So we did functional analysis, and we analyzed the function of the vessels. And Pierre Lacombe in the lab analyzed two key properties of brain vasculature. So he analyzed the autoregulation of the cerebral blood flow, and he analyzed the functional hyperemia. This is an adaptive response, which means that when you have a neuronal activation, you have an increase in the CBF. So he analyzed the CBF autoregulation, and what he found is that in the mutant mice, the autoregulation curve, at least the lower, the left part of the autoregulation curve, this curve is shifted to the right, so suggesting that even with a minor decrease of the blood pressure, the cerebral blood flow starts to decrease. And when you stimulate, so the paradigm in this experiment to analyze the functional hyperemia, you produce a mechanical stimulation of the whiskers in the mouse. So you stimulate the whiskers and you record the cerebral blood flow on the top of the somatosensory cortex. And in wild-type mouse and the controls, you get a 55% increase in the CVF, and this increase is attenuated in the mutant mice. So there is something wrong in the brain vasculature, and which occurs prior to the occurrence of brain lesions, since we can detect these abnormalities at 6 months of age. So to further go into the mechanisms of at the level of the vessels, Céline Baron in the lab, she isolated the brain vessels and she analyzed the myogenic responses. And so she mounted the brain vessels onto pipettes. She applied transmural pressure, and myogenic response means that when you apply pressure and you increase pressure step by step, the vessel should at least maintain its diameter or even constrict. And what she found is that if there is a decrease and that the myogenic responses are attenuated. And Fabrice Dabertrand in Mark Nelson's lab, he isolated intraparenchymal arteries and found exactly the same result, indicated that the mutant mice, both in France and mutant mice in the United States, have the same defects on the large vessels and on the very small vessels. So definitely there is something wrong at the level of the vessels in these mice and prior to the occurrence of white matter lesions. So I won't go into the details of the other findings. So Marie in the lab found that there is, from 12 months of age, an alteration of a capillary bed, and Pierre Lacombe measured the resting CBF, and he measured at 12 months, 20 months, and he found a global chronic cerebral hypoperfusion. And recently, Carmen in my lab found some evidence of decreased cerebral blood flow resting CBF as early as 6 months of age. So what we learn is that the earliest vascular changes is a cerebrovascular dysfunction. But at this point, we know that this is the earliest cerebrovascular earliest changes, and we have not demonstrated that this change causes this. And I think it's a key issue for the next year of research. So to move to the second question we try to address is the disease caused by a malfunction of a receptor? So is the disease caused by a loss of function of the receptor? So very simple question, but very debated answer. And the answer is a bit more complex. So let me first explain what is the normal function of a NOD3 receptor in the brain vessel. So during the past years, using a various mouse model, we demonstrated that NOD3, which is predominantly expressed in the vessels, and especially in the brain vessels, that NOD3 plays a critical role in the brain vessels, and in the absence of NOD3, to make the things very simple, in the absence of NOD3, arteries look like veins. both at the histological level and at the functional level. So we have an alteration in the maturation, the postnatal maturation of smooth muscle cells, and we learned that smooth muscle cells need to undergo a postnatal maturation to remodel the cytoskeleton, and we have an alteration of a differentiation, of the arterial differentiation, because smooth muscle cells within the wall of an artery have an arterial set of genes. And recently, in my lab, Singer-Polignac identified, so the NOTCH3 receptor, like the other NOTCH receptor, is expressed at the plasma membrane as an heterodimer, as Elizabeth showed. And when the receptor is activated and bind to ligands, transmembrane ligands in the neighboring cells, it triggers a number of cleavage, allowing the intracellular domain to translocate to the nucleus, interact with the transcription factor or BPG-kappa, and activates the transcription of genes. And Singer-Polignac has identified a number of these genes which are regulated by NOTCH3, and among these genes there is NOTCH3, so NOTCH3 regulates its own expression, and it regulates some genes which are important for the myogenic responses, like KV1.5, and also GRIP2, for which we showed a role in the myogenic responses. So the key question is, the key question is, what do the mutation do on this function? So we have very stereotyped mutation. All the mutations lead to an unknown number of cysteine residues. So the expectation was that the answer would be very simple, but the answer is complex. So definitely, a few mutations located in the ligand binding domain, which is constituted by EGF receptor, EGF repeats number 10 and 11, a few mutations located in this region, definitely there are loss of function mutation. So with this mutation located in this region, you get a dead receptor. And this is illustrated on this slide. On a direct manner, we used a transgenic reporter in the mouse, which expressed the LAC-Z protein, which produces this blue staining in the vessels, and which is under the control of the responsive element of RBPG. So in wild-type mice, you have this blue staining of the brain vessels. In NOTCH knockout mice, so the staining has gone away. And when you reintroduce a wild-type NOTCH3 protein, you rescue the blue staining, you rescue the activity, and when you reintroduce a protein with this mutation in the ligand binding domain, so you do not rescue. And we use multiple assay, and you can believe me, this mutation in the ligand binding domain lead to a loss of function. However, the key question is, does this apply to all the mutations? So do all the mutations, in the end, result in a loss of function? And I would like to show you some recent evidence that we obtained to show you that the answer is no. So we collaborated with Mieke de Verschijn in Belgium, and Mieke has generated these mice with this point mutation, exactly the same mutation of the first mouse model I showed you. So it's a very clean mouse model because he introduced the point mutation, so it's exactly the same situation that in patients. You have a point mutation, no overexpression. And the only straightforward approach to monitor NOTCH3 activity is to measure the level of NOTCH3 target genes. And we established a toolbox which allowing to measure the NOTCH3 activity, and we made sure that this toolbox was able to detect an increase, a decrease, and a decrease as low as 50%. And this is only a subset, so in these mice we first showed that these mice at the heterozygous state developed the NOTCH3 CD deposits. At the homozygous state, they developed much more NOTCH3 CD deposits. We collected the vessels, we dissected the vessels, and we quantified a number of NOTCH3 target genes, and this is only a small subset of genes, and definitely there is not any alteration. So strongly suggesting that this mutation, even at the stage where there is an accumulation of NOTCH3 CD, no alteration of the activity. So to make sure, we did another experiment. So it could be that with the progression of a disease, with the accumulation of a protein, the mutant protein loses its activity. So we went back to our initial mouse model, and our initial mouse model has been done on a wild-type NOTCH3 background, and the expectation is if a mutation induces a loss of function, if we remove the endogenous NOTCH3, we should get a more severe phenotype, and this is the experiment Emmanuel has done in the lab. He has produced these mice, the mouse model on the NOTCH3 null background, and he examined in detail the white matter. So first, in these mice, the NOTCH3 null mice don't have any white matter lesions in this paradigm at this age. So these are 20 months old mice. So maybe if these mice were a little bit obese or had some hypertension or smoke, or so in this case, so these mice do not develop white matter lesions. And the phenotype of our CADASIL mouse model is not more severe on a NOTCH3 null background. So strongly suggesting that reduced NOTCH3 activity, even though some mutation can lead to a reduction of NOTCH3 activity, so reduced NOTCH3 activity is very unlikely to be the driving force in CADASIL. And this result is further supported by a recent publication from the Dutch group who identified a patient with a compound heterozygous and carry a pathogenic mutation on one allele, and on the other allele he has a deletion of NOTCH3, and this patient is not more severe. So at this point, we think, I'm not saying that the absence of NOTCH3 will not produce any phenotype, and obviously Elizabeth showed you this morning that patients with a loss of function mutation could have a phenotype. I'm not saying that it does not produce any phenotype, but in the case of a pathology of CADASIL, so the absence of reduction of NOTCH3 does not play any role, it does not play a major role. So it's not the driving force. So what is the driving force? So the common denominator to all the mutations is the presence of vascular deposits of NOTCH3, ECD, and GUM. So first, what is the relationship between the accumulation of NOTCH3, ECD, and these GUM deposits? So we published, in 2000, we published, using an electron microscopy, immunoelectron microscopy analysis, we published that NOTCH3 accumulates, so at the edge of the GUM deposits, and we said, so we can also be wrong. Because we published in 2000, we published that there was an accumulation of NOTCH3 CD at the edge of the GUM, but not within the GUM, and we were wrong. So using a more sensitive technique, a more sensitive approach, our lab and other lab found that there is some NOTCH3 within the GUM deposits. But maybe the truth is in between. So it's possible, so the GUM have a mushroom shape, and it's possible that the concentration of NOTCH3 CD is not exactly the same at the edge versus the head. And what we usually find is that there is more NOTCH3 CD at the base of a GUM and less NOTCH3 ECD within the GUM. Anyway, there is NOTCH3 ECD within the GUM. So one mystery in the accumulation of NOTCH3 is that we only have an accumulation of the extracellular part of a receptor. We don't have any accumulation of the intracellular part, and we use multiple approach. So the current model posits that there is a dissociation of a receptor upon ligand activation of a receptor. So it has been suggested that we had an accumulation only of the ectodomain because the accumulation occurred after the activation of a receptor. So we did this very simple experiment. We looked at the NOTCH3 ECD deposits in both patients carrying this point mutation which lead to a dead receptor, and also in the mouse model. And what we found in both patients and in the mouse model, there are deposits. So definitely the accumulation of NOTCH3 ECD occur independently of ligand activation of a receptor. So why there is only an accumulation of the extracellular domain? So at this point, I can only make some speculation. So we know that receptors recycled from the plasma membrane to the intracellular, to the cytoplasm, recycled, and it could be that during recycling, when they are in vesicles and endosomal vesicles, the intracellular domain can be degraded and the extracellular domain cannot be degraded and goes back to return to the plasma membrane. So we then examined the mechanisms of formations of these deposits. And to do this, we first tried to determine the biochemical correlate of these NOTCH3-ECD deposits. And we used the following approach. So we took advantage of our various mouse models, and we took advantage of the tissues, post-mortem tissues, and definitely we absolutely need post-mortem tissues from patients. So we dissected the brain arteries from our mouse models, and we isolated the brain vessels from frozen material from brain samples of patients and controllers. And we did fractionation experiments, and what we found, this is a Western blot, we found that the NOTCH3-ECD protein accumulates, so it is present, it does not accumulate, it's present in the soluble fraction in the wild type mouse model. In the mutant mouse, in the mutant mice, there is a huge accumulation of a NOTCH3-ECD in a very insoluble fraction. And whatever the detergent, the denaturant you use, it's in this insoluble fraction. You can only solubilize the NOTCH3-ECD in the mutant mice, and we got exactly the same results in tissues from patients, only by adding a reducing agent. So strongly suggesting that there is an accumulation, so that the NOTCH3-ECD forms multimers, and that these multimers are mediated and are involved disulfide bonds. And this is very consistent with in vitro results from Dishgan's lab, showing using a very small recombinant NOTCH3 proteins containing the first 5 EGF repeats, and he showed that this small fragment can form multimers, and that these multimers involve disulfide bond. So we next determined the proteome of this fraction. So if the gum deposits are composed, so we reasoned that in this fraction we should find the proteins contained within the gum deposits. So if the gum deposits contain only NOTCH3-ECD, we should find by proteomic analysis only NOTCH3. So we collaborated with Vince's lab in Paris, and we determined the proteome using MSMS analysis. And we found, obviously, NOTCH3-ECD as a positive control. And we did this experiment, this mass spectrometry analysis. We did these experiments. We use human tissues and we use murine tissues. And the idea of using murine tissues was to get a full repertoire at the end stage of a disease. And the idea of using murine samples was to pick up the proteins at a very early stage of a disease. And we match the two lists. And within these two lists, by combining these two experiments, we ended up with a very short list of proteins which accumulates in the mutant vessels. And within this list, we found TIM3. So again, the number 3, so it's a pure hazard, I think. And TIM3, so it's tissue inhibitor metalloproteinase 3. And we first validated the MSMS results, and we found that there is obviously an accumulation of TIM3 in the mutant arteries from the mutant mice in this fraction, which is highly enriched in NOTCH3-ECD, and we could validate the results as well in the brain vessels. So brain vessels isolated from post-mortem brain samples of patients, and there is an accumulation of TIM3. So we could validate as well by immunohistochemistry and in patients only, because unfortunately there is not any good antibody in the mouse. So there is an accumulation of TIM3 in the brain vessels of patients, and these accumulations co-localize with NOTCH3-ECD, and there is some TIM3 in the gum deposits. So TIM3 is a component of the gum deposits. So we picked up also vitronectin, and we did the same experiment. So as again, vitronectin accumulate and produce this, the staining produce this very characteristic immunostaining pattern with these granular deposits, so both in the mice and in the patients. And the finding of the accumulation, this granular accumulation and these deposits of TIM3 and vitronectin in the mutant mice indicate that it is probably a very early, we are at the very early stage of a disease, that it occurs early in the disease process. So we next investigated the mechanisms of why TIM3, why vitronectin accumulates, why the NOTCH3CD promotes the accumulation of TIM3 and vitronectin. And we used in vitro approach, and as you can see on this co-immunoprecipitation experiment, in smooth muscle cells which physiologically express endogenous level of TIM3 and NOTCH3CD, there is a teeny interaction between NOTCH3CD and TIM3. When you immunoprecipitate NOTCH3CD with an antibody against NOTCH3CD, you pull out a very small amount of TIM3. But when you overexpress TIM3, you can pull out a huge amount of TIM3. So indicating that this is the excess of NOTCH3CD or the formation of aggregates which promote the accumulation with TIM3. And this is mediated by the extracellular domain, because when we repeat the experiment with a NOTCH construct which is deleted of the ectodomain, we do not pull out TIM3. So does the mutation interfere with this interaction? The answer is no. And it's the excess of NOTCH3CD or the aggregated form of NOTCH3CD which promotes the accumulation and the interaction with TIM3. And also in vitro, we saw that when we overexpress NOTCH3, we promote the accumulation, we favor the accumulation of TIM3, and again, this is an effect which is mediated by the extracellular domain. So the excess of NOTCH3CD promote both the interaction and the accumulation of TIM3. And what about vitronectin? For vitronectin, the story is interesting because vitronectin does not interact with NOTCH3CD, but vitronectin interacts with TIM3. And when we put all these three proteins, TIM3, vitronectin, NOTCH3CD, we see a ménage à trois. So, suggesting the following model for the formation of the deposits. Initially we have a wild-type receptor and a mutant receptor. So the first step, at the first stage, we have an accumulation of NOTCH3 ECD and formations of multimers. But once these multimers have reached a given size, it can promote the accumulation of other proteins like TIM3 or other proteins, and then these proteins can trigger the accumulation of other proteins, so TIM3 can probably promote the recruitment of vitronectin with a snowball effect. And this is very consistent with what we see in various mouse models, where first NOTCH3CD deposits and then the gum deposits. So the key question is, are these proteins which accumulate simply a marker, or does it contribute to the disease process? And this is the next question we addressed. And I would like to share with you some recent results of Carmen Capon, who recently joined my lab. And we used the cerebrovascular dysfunction as a phenotype, as a paradigm to address this issue. So what Carmen showed, so to address this issue, we used a genetic interaction approach, and we first examined the contribution of the accumulation of TIM3 in the disease process. So we used a gain-of-function approach on first, and so just to remind you some key features of TIM3. So TIM3 belongs to this family of TIMP protein, and TIM3 is the only TIMP protein which bind to the extracellular matrix. So all the TIMP proteins can inhibit the MMP, the metalloproteinase, and TIM3 has some specific function. It inhibits the ADAM17, which is also called TACE for TNF-alpha converting enzymes. And interestingly, there is a genetic disease affecting the eye, and it's considered more or less as a Mendelian form of age-related macular degeneration, in which there is an accumulation of TIM3. So we generated first a mouse model, a new mouse model, which overexpressed TIM3, and we used a large DNA fragment to overexpressed TIM3 with the same endogenous expression pattern. And we got 2 lines. And this line overexpressed, it's a western blot with the brain arteries. And this western blot shows you that we have an overexpression which we quantitated at about 6 times over the non-TG mice. So what Carmen did, on first, she established the paradigm using an open cranial window, and she looked at the cerebrovascular response using this open cranial window paradigm. And on first, what she showed is that in the mutant mice at 6 months of age, we have an attenuated responses to whisker stimulation. These are the controls in the black bars. The blue bars are the responses to the cadazine mice. So we have an attenuation to the whisker stimulation, the responses to whisker stimulation. We have also an attenuation of the responses to acetylcholine topical application and an attenuated response to adenosine, topical application of adenosine. So in this case, we look at the endothelium-dependent response, and in this case, we look at the smooth muscle cell quite dependent responses. An interesting, just a brief comment, Carmen found that these responses were attenuated at 6 months, but were not attenuated at 2 months. And at 2 months, we have natriuretic ECD accumulation. So what did Carmen? She looked at the responses in these mice, in this mouse model, and she found that the defects were exactly similar to the cadazine mice. But it's not a proof. So we did the opposite experiment, and we reduced the amount of TIM3 in the cadazine mice. And the expectation is that if the accumulation of TIM3 contributes to this phenotype, we should rescue the cerebrovascular dysfunction in these mice. And what we would jump, yeah, I'm finishing. So she did the rescue experiment, and what she found very interestingly is that in the cadazine mice with a reduced amount of TIM3, the cerebrovascular dysfunction is completely rescued. And to further establish to further confirm, Céline Barréon did exactly the same response, did exactly the same experiment. In this case, she looked at the myogenic responses, and she found that the myogenic responses, which are attenuated in the cadazine mice, are completely rescued by reduction of TIM3. So establishing that TIM3, the accumulation of TIM3 contributes to the disease phenotype. So I will skip these slides, but just to show you that we have additional evidence and pharmacological evidence that this effect is mediated not by an over-inhibition of MMP, because when Carmen superfused when she superfused TIM3, she can get exactly the same result. And when she superfused either TIM2 or TIM1, there is not any alteration. And I will go very fast, and very surprisingly, this effect is mediated by an over-inhibition of TACE, and just a simple topical application of TACE in the cadazine mice can partially rescue the deficits. So to conclude, and this is one very last slide, to conclude that at this point, we propose this model regarding the mechanisms of cadazine. So we propose the natriuretic ECD cascade hypothesis. It's not very original. It has some analogy with the amyloid beta cascade hypothesis. And in our hypothesis, I would like to point out that what we think at this point is that the natriuretic ECD deposits are very important, and the trigger, we speculate that this, at least we have some evidence for TIM3, that these deposits will trigger alteration of the matrisome, so the extracellular matrix proteins of the matrisome. So the proteins which are located between smooth muscle cells, so smooth muscle cells are in direct contact with the astrocytic end feet, and we think that the problem is there with an abnormality of with the proteins of the extracellular matrix in this region. And we have demonstrated that TIM3 contributes to cerebrovascular dysfunction. We have some evidence that TIM3 does not contribute to the alteration of the capillary bed, so we think that we have a multifactorial toxicity. And TIM3 could contribute at the later stage of a disease to the pathological alteration, so thickening of brain arteries, given its role on MMP. And in the end, that this alteration, we think it promotes an energetics failure. And what I mean by energetics failure, it's a failure of the ATPase-dependent pump, which will impair ionic homeostasis. So finally, I would like to thank members of my team. So Emmanuel Cognac, Carmen Capone, Céline Baromangui, and a very special thank to Valérie, who has done a wonderful work for many, many years and who is trying to survive, working with me for many, many years. And I would like also to thank the past members of the lab, and especially Pierre Lacombe and Marie Monet. And I would like also to thank the many collaborators during these past years, and especially members of the Duke Network. And I would like also to thank the brain banks and the facilities, and also our funding agencies. Thank you very much.